Next important point we are going to discuss is the purpose of sampling. So why do we need to do the sample? Okay, like sample always will have some flaws. If there is a population of 50k, I wanted to draw conclusion about 50k by collecting the information from 1000. So the 1000 may or may not represent the entire population. Okay, but why we do that? Now the question is why we do that? Because there are always the chances of variation. Population uh, characteristics may be different from the simple characteristics. Okay, it is not a 100% foolproof method, but still we do it. Why? To make an inference about the population. Why we need a sampling? Because we want to make an inference, means we want to conclude. We wanted to know about the population, and population is infinite. It's very difficult to go everywhere to each and every person, every element. So we can collect the data from a small representative. We call that sample, and we come to know about the population. Studying entire population is impractical and impossible, which already I told you. Sampling in research save a lot of time and labor. Because if you can get the same, almost similar amount of accuracy with a small sample, why we should go for a complete enumeration? It is cost effective because it minimizes the cost. If you collect data from 50,000, it will take a lot of time, it get involved a lot of money and many more things. Tasting of destructive sample, like for example milk. You want to describe the quality of a milk or your blood. So if I wanted to know the HP percentage, HP of your blood, can I withdraw the entire blood from your body? No. I have to take only a small amount of blood, right? So for destructive sample, like if you want to check the quality of a milk, you cannot put chemical in entire lot of milk. You can just take a small sample. Provide accuracy and organize the convenience. Now statistical tests are so strong, so powerful. They will give you approximately, results with a similar accuracy as if you collect data from the large population or entire population. If collecting data from a 50,000 is giving me a 99% accuracy, and collecting data from 1,000 sample is giving me, let's say, 98% accuracy, I should go for the 1,000 samples, right? Then control the unlimited data, because if you are involved entire population, which is practically not possible, and you have to deal with the data that is very extensive and will not be manageable. Give a greater speed, help to complete in stipulated time. So of course, if you go for a sampling, it saves your time and you can complete your research project in time. Assist to collect the intensive and exhaustive data. See, your purpose is to collect the information. If you're collecting the information from a small sample or an optimum sample, okay, optimum sample means the sample that is required for that particular variable. We are not going for very large sample, neither going for very small sample, but the optimum sample required for that. So that is also we can more focus on the information. So these are the purposes of sampling. Now there is another important concept here, sample versus complete enumeration. Okay. So sample, as I already told you, sample is a part of population. The information is collected from only few units of population and not from all the units. Okay. So that is a sample which already I've told you. An advantage, it is more practical and be feasible. What is complete enumeration? It is also known as a census. Full enumeration means you collect data from each and every element of the population. Like in the previous example, if there are 50,000 nursing students and you've taken data from 1,000 only, 1,000 what? They represent the entire population. That is a sample. But if you go to the all 50,000 and you wanted to know about the same thing, then it is a complete enumeration. Okay. So under this, the information are collected from each and every unit of the population or universe. And advantage, of course, you get exact information with a higher accuracy. If I've taken data from the entire population, it will be more accurate than the sampling one. Now, what are the criteria for a good sampling? So, good criteria for a good sampling, it should be homogeneous. The selected sample from the population should be homogeneous and should not have any difference when compared with the population. See, I'm taking the subset of the population only. So they should represent the population. Suppose there are 50 students in a class and I have taken 5, and these 5 students are having a 98% in a previous exam, whereas the remaining 45 people are having an average is 60%. That means they are not representing the entire class. I have to take the sample which are homogeneous, means which are similar to the population. Then only I can draw the conclusion about the population. Optimum sample size. Another important point is optimum sample size. Okay, reasonable number of items to be included in the sample to make the result more reliable. It is not the large sample. It is the adequate sample that required to desired power. To desire power of what? Power to determine the relationship among variables if exist. Usually, if you take a very small sample, it will give a non-significant results. It will fail. It will reduce the ability of any statistical test to determine the relationship if it exists. If you take a very large sample, it will waste a lot of resources. So the concept of optimum sample size, reasonable number. Then true representative, which already I told you, it should be homogeneous and is mutually exclusive. That is another important point. One sample should not influence the another sample. That is another important point. The individual items composing the sample should be independent from each other. Like for example, you have taken a sample of 5 people, and among 5 there 2 are the twin sisters. Okay, they may influence each other's. Their opinions may be same, their IQ level could be the same, they're coming from the same home. So they are not, we cannot say they are independent. Their opinion may be dependent on each other. Okay, so whenever you select a sample, the sample should be independent. So these are some of the important criteria that one has to consider while doing this. Next important point that we need to understand here is sampling for quantitative and qualitative researches. Already we have discussed in detail about the quantitative and qualitative researches. Now, how the sampling, vary, and what are the points that we need to consider while taking these kind of researches. If we talk about quantitative researches, here what we do, we often select sample that will allow achieving the statistical conclusion validity. What is that? Desired results, or what I say, statistical conclusion validity means we take the sample in the reasonably large number which determine the relationship if exist. Let's say, if suppose I want to establish the relationship between the knowledge and gender. Now, knowledge is the outcome variable, it's a qualitative variable. Now, I need to, if suppose I have taken data from 30 people and I'm saying there is no relationship between knowledge and gender, means knowledge is not influenced by gender. So one possible reason could be because of small sample size. Because sample size, if reasonably low, it will reduce the power of test. Already we have discussed what is power of test in our statistic classes. So you can refer that particular chapter or the video lecture. So that cannot be done. Now suppose how much should be the sample size to determine the true relationship among the variables. That we call a statistical conclusion validity. If there is a difference, like let's say male are having a mean knowledge score is 20 and females are having mean knowledge score of 19. Yes, there is a difference. You can say the males are having a more knowledge. But in statistic, can we say statistically they are different? Something is different has to be tested. Whether the difference is an error difference or it is a statistical difference. And to determine and to have a statistical difference, you need to take a large sample size. So that is a one important point one has to consider, please. Second point is to generalize the study. There is another important purpose for sampling for quantitative research is to generalize the results, okay? Because mainly your focus is to generalize. So whenever your focus is to have a generalization of findings, you need to collect data from the large sample, okay? Determine the sample size in advance. That is another important thing that we do. We decide the sample size in advance for quantitative researches. Use both. We can use probability and non-probability sampling techniques for this particular type of design. So usually you see some of the researches, quantitative researches use probability sampling where they use the random methods, and some of the researchers use the non-probability sampling where they use the like purposive sampling. They select by their based on their convenience and all. Now, what is the picture here about qualitative researches? Sampling in qualitative research. Number one, to develop a rich holistic understanding of the phenomena of interest. Here, the purpose is not to determine a statistically valid results. Here, the purpose is what to understand the phenomena. So, if you understood a phenomena by collecting data from a 3 sample or a 5 sample, you will stop it there only. Okay, and the purpose is not to generalize, the purpose is to transferability. Can I transfer these results to some other population, or after doing that will I get the similar results? Like one of my qualitative research where we identified that the children engaged in child labor are having a more autonomy, they feel more autonomy. So now the question is, can I transfer these results to some other children engaged in child labor? That is the point, okay, not the generalizability. They make sampling decision during the course of data collection based on the information. So usually we don't decide that in advance. What we do, we collect the data, we analyze it. That is what we call a ongoing analysis. And collecting the data from the new sample or next sample, we decide based on the analysis of the previous data, and something we call saturation. Okay. So if someone asks you what should be the sample size for qualitative researches, you say it is not decided in advance. It is based on the saturation. Saturation means where you collect the data until you start getting the new information, and there will be a point where you will start getting the repetitive information, and there the researcher has to stop the data collection. Sample size till data saturation, and use the, usually they are non-probability. Okay, non-probability sampling are the methods. So please remember the difference. There in quantitative research, what we do, we focus to determine statistically valid results. Here the purpose is just to understand the phenomena. In quantitative research, sample having a purpose to generalizability, that's why we have a large sample size. Here the purpose is to see the transferability, how the results stand true if we transfer to some other population. Here we have advanced planning of a sampling, like what techniques, what sample size, all that. But here decision is being taken in the ground itself, how many samples are required. Here sample size we decide in advance, and here we follow the data saturation principles. And lastly, we use both type of methods like probability and non-probability for quantitative researches, and we use non-probability for qualitative research. Let's talk about briefly about the various principles of sampling. These are the points which provide us the basic understanding and theoretical understanding of sampling. Now, the three principles, there are many more, but I am just focusing on these three. Principle of statistical regularity, principle of large numbers, and principle of optimization. Though all these principles are very much connected with each other, and if you look at the basic understanding of these principles, it will take you towards the one common end. Okay, so you can conclude in that way. Let's talk about that. First one, principles of statistical regularity. What is that? This law is based upon the mathematical theory of probability. Okay, so this theory believes that there are chances of happening of any event, and we can calculate that chances, or what we call the probability, okay, by applying some of the mathematical calculations. Okay, so what this theory says, it is based upon the following two major conditions. One is a large number size. If you really want your sample should represent truly to your population, then you should follow this principle, the principle of large number. What is that? As the sample size increases, the true characteristics of the population are more likely to reveal. What I am saying, suppose you wanted to know the HB in a specific class, there you assume to have 50 students, and if you have taken data from 5 students, and let's assume that the hemoglobin is around 11.9 of these 5 students. No, these 5 are the sample of these 50. Now instead of 5, if you take the 10 sample from the class of 50 and you see that if the hemoglobin level is 12.1, that means it is more near to that. No, because it's a like finite population. If you know the actual hemoglobin level of this class, suppose the actual hemoglobin of class is 12.4. Now as you see, as you increase the sample size, suppose now from 10, if you taken another 5 more sample, 15 sample, this will go very near to the real characteristics of the population. So in simple term I say, if you increase the sample size, you're more likely to represent your population. Second principle here is a random selection. A sample should be selected randomly in which each and every unit of universe means population, has an equal chance of being selected. So please understand that point. What this is? Random selection. It is desirable under the principle of statistical regularity. If you really want that your population inference, which is based on the sample, should be valid or accurate, you need to select this sample randomly. What is random? Random means where every unit, every subject or a sample is given an equal chance of being selected. You can adapt from the various methods of probability like simple randomization, or you can have stratified or cluster randomization, but everyone should be given an equal chance. Like for example, from the group of 50, I have to draw the sample of 10. How I do that? I can just put the name of all these 50 and I can just draw. I can have a drawing from that. Okay, so all the 50 chits which I'll put somewhere, and from there I'll select the 10 randomly. So that is the principle that is desired by this. If we talk about the second principle, that is a principle of large number, which already I've told you. It is based upon the concept that as the sample size increases, the better the result you are going to get. So as I say, if you're from 50, if you're taking a sample of 5, suppose assume the population HB is 12.4, 5 sample is giving you 11.9. So this is a difference, na? Actual population hemoglobin level is 12.4, but the sample of 5 is giving you 12.9, sorry, 11.9. So this is the error, na? So if you want to minimize this error, what you can do? You just increase the sample size 10 or maybe the 15. So you'll become very close to the actual number. So that is a principle of large number. So your result would be more valid, okay? And that is why whenever you read a paper or whenever you look at some researches, you should see how many samples they have drawn. Okay, next is principle of optimization. What is that? Now this is a slightly different concept than the large number. In the first two principle of statistical regularity, where also where we have talked about large sample size. In a second principle also says large sample size, means if you have a large sample size, you more likely to get the more accurate results. But what is this principle is all about? One must be able to get optimum result with a maximum efficiency with a minimum cost. That is the principle that one need to understand. One side it is clear. If there are 50 subjects, you draw data from 50. Fine, you will get more accurate results. But you are taking 5 sample, less accurate. You are getting 10 sample, bit accurate. So your accuracy will increase as you increase the sample size. But the point is that let's say if you have taken a 40 sample and the hemoglobin of this 50, if I say is 12.4, and the hemoglobin that we could estimate from the 40 sample is, let's say, 12.3, and if you've taken only 20 sample and this hemoglobin level is 12.1, so now you have to decide as a researcher, you are getting almost similar accuracy, but as you say, Here the sample size is almost double. After collecting data from a 40 sample, you are getting accuracy that is slightly similar, almost similar to the sample that you taken only from 20. So that is the principle of optimization. You cannot go always for a large sample. You should not think about that. You should think about the optimum sample. What is the optimum sample? You get a maximum efficiency. Your results are more precise, but with a minimum cost. As you increase the sample size, that sample size will increase your cost as well. So you have to maintain a balance in between these two things. So these are the three important principles of sampling. Next, we'll talk about another important point here, just steps in sampling process. Okay, sometimes they ask this question in exam in a 5 or 7 marks. What are the steps one has to follow while deciding a sample or by selecting the sample? So very simple principles. The first one is identify the target population, which already I have told you what is target population. It is the overall population, group of people. From there you are going to generalize your findings. Okay, there you are going to generalize your findings. Okay, and of course a researcher needs to identify the population of interest based on the research topic. That is a very important. If suppose you wanted to collect the data regarding the prevention of HIV/AIDS among truck drivers, so truck drivers become your target population. Okay, second is identify the source population. Accessible population is also known as a source population. Now all the truck drivers in India are your target population. Now you practically it is not possible for you to collect the data from all the drivers, okay, from all the states, from all the locations. So what you can do, you have to decide about the source population. So the accessible population is the group of individual or object from which the sample might be taken. So you have to decide. Now see, suppose you have decided the people, those who are truck drivers, those who are visiting to the national highway, selected national highway, let's say national highway number 8 or national highway number 1 or 52, okay, all that. So you've taken some of the area, and from there those the drivers, truck drivers, okay, just traveling through these routes will be taken as your source population. Okay, so first you identify your target population, from there you select your source population, then select the sampling frame. That is another important point. Now this is not a mandatory step for all type of sampling techniques which we are going to discuss in subsequent lecture, but this is required if you are doing a probability sampling technique. If you remember, I told you in a previous lecture that sampling frame is nothing, it is a listing of all the prospective participants. Okay, so in the same example, all the truck drivers, those who are visiting or traveling through these national highways will be taken their list. You have to prepare the list of all the people traveling there, all the truck drivers, and suppose there are 1000 drivers are visiting through these routes in a defined time. So that 1000 name of all these 1000 truck drivers become your sampling frame. So sampling frame is a list that you have to list all the people from your source population. What is next? Then sample eligibility criteria. Once you have a list of people, then you have to decide, and even before that you should see how many of these people are as per the eligibility criteria of your research, which already we have discussed. What is eligibility criteria? These are the characteristics which make someone eligible to participate in your research, or maybe based on that the specific prospective participant may not be able to participate in your research. Okay, so just two basic criteria which already I have told you. You should see who to be taken inside the research or into the research, that we call it inclusion criteria. Okay, like for example, you decided I'll take only the people having an age between truck drivers between 18 to 60. So anybody above 60 will not be taken. So that is your inclusion. You will only include the people having age 18 to 60. Then you decide I'll take only the male. Then you decide I'll take only those who are having a DL, driving license, a valid commercial driving license. Okay, so these three criteria, that is your inclusion. Now from there, you suppose you said, okay, somebody is having an age of 18 to 60, male, or having a driving license, but I will exclude those who are not willing to participate. Not willing. Suppose out of 1000, Okay, you suppose there are 800 people who are found to be eligible to participate in your research, and from that suppose 20 people says that they won't participate, they're not willing to participate. So your final sample would be, you just do that. Okay, so 780 would be your sample. Okay, next is specify the sampling technique. Once you decide on that, next important point is you should decide what sampling techniques. The technique means it is a process that you adapt to select the sample from your sampling frame and from the population. So sampling can be done through probability, that is a random sampling, as well as from the non-probability sampling. So you decide that whether it's a random or non-random. We'll discuss that particular point in detail in subsequent lectures. Next important point here is to determine the sample size. Now, how many samples are required? For that also there are so many different ways are available to decide the sample size. So sample size is simply the number of units in the sample. Sample size determination depends on many factors like how much time, what cost you have will get involved in selection of sample, what facilities are available. In general, large samples are better, but they also require more resources. So please understand that always follow the principle of optimum sample size, neither too small nor too large. You should maintain the high efficiency or validity of your research, but at the same time you should also keep your cost at the minimum. Then execute your sample plan. Okay, so finally you've decided all these things. So once all above stated steps are completed, researcher can use all that information to choose sample for the research study. So these are the 7 important steps that one need to remember. I hope you will remember these steps. So if I say first you should identify the target population. From there you make your decide your source population. Then make your sampling frame. Again I say sampling frame is nothing, it is a listing of all the peoples. Then decide your sample eligibility criteria, means the characteristics that make somebody eligible to participate in your research. So we have a 2 here, inclusion criteria and the exclusion criteria. Decide about the sampling techniques, what specific techniques you are going to use to draw the sample from the population. Primarily there are 2 basic techniques. We have that probability and the non-probability sampling technique. Decide on sample size, how many units, how many samples are required to get a desired results. Okay, and finally execute your sample plan. Whatever you decide, please plan accordingly. So remember these 7 steps of this. So that's all about the principles and steps involved in a sampling process. Thank you.